In this video, I will be discussing how gel electrophoresis is used to separate DNA fragments of different sizes. So let's say you have a sample that has a bunch of copies of this piece of DNA, and you use a restriction enzyme to digest this molecule into two pieces. Here is the restriction site, so we cut here and create a small fragment of DNA and a larger fragment of DNA. Now I want to be able to isolate this fragment to use later in my lab. Right now, I just have a sample containing some medium-sized pieces mixed in with some smaller-sized pieces of DNA. I can use gel electrophoresis to separate all of these medium-sized molecules from all of these small molecules. How do I do that? So this procedure uses a gel, usually made of agarose, that is placed in a buffer that has ions that can carry current. The gel contains indentations called wells, where we inject the DNA sample that we want to separate. We then apply an electric field that has a negative pole on the side of the gel with the wells and a positive pole on the other side of the gel. We should remember that DNA is negatively charged due to the phosphate groups in its backbone. When the electric field is applied, the current is running through the buffer. The negatively charged DNA fragments move away from the negative pole toward the positive pole through the agarose gel. In this process, the larger the DNA fragment, the slower it moves, causing the fragments to separate based on their size as they travel. In this case, let's run two samples through our gel. In the first well, let's run an undigested sample that only contains the full large segment of DNA that has not been cut by the restriction enzyme. In the second well, let's run the sample that has been digested by the restriction enzyme, resulting in this medium-sized fragment and a smaller fragment. As the gel runs, the smaller fragment will move faster through the gel than the larger fragment, and we end up with something that looks like this. In the first lane, we can see that our large undigested molecules do not move very far in the gel. In the second lane, we can clearly see two separate bands of DNA. We have successfully separated these two different sized fragments, the medium here and the small here. And we can cut either one of these fragments out of the gel to use the DNA in further lab protocols, perhaps by inserting it into a plasmid and transforming some bacterial cells. So that is gel electrophoresis. We load DNA fragments of different lengths into a gel, and then the smaller ones move more quickly than the larger ones when current is applied. If you want to learn more about how you can digest DNA using restriction enzymes, see my video on restriction enzymes. If you're interested in how you can insert isolated DNA fragments into a plasmid and then transform bacterial cells, see my video on transformation.